I am very, very honored to introduce our speaker, Leo Cohen. So Leo and I were down in Taipei in May of 2013 as we were talking. I said, you know, we should invite you up. And so this is a promise of the fulfillment of that promise. So Leo is the chief of Human Cortical Physiology and Neurorehab section at the NIH. Leo is world-renowned in non-invasive brain stimulation. And we wanted to pick his brain, no pun intended. We had a great dinner yesterday, and just, we didn't talk much about science, but we had a lot of fun though. So Leo, enlighten us, come on up. Oh.
Thank you, John, for inviting me. I hope I'll give you some food for thought on some issues that are of common interest. My background is in neurology, and I have always been interested in how the brain reorganizes to compensate for loss of function, particularly after a stroke. And so the strategies or the tools I have been using to study these forms of brain reorganization include functional neuroimaging and electrophysiological techniques like EEG or MEG. And I have been trying to study mostly at the proof of principle level strategies that could enhance what is the gold standard in neurorehabilitation, which is training. So the types of questions I have been looking into are how can we improve training effects in neurorehabilitation? From a strategic point of view, I have done a number of studies in healthy subjects, and whenever some indications or potentially instructive implications emerged from those healthy volunteers studies, I try to move into small trials in patients with stroke predominantly, and when meriting to go forward, then some of those trials and the fellows who carried them out in my lab moved on to carrying out larger trials outside NIH. So some of the work that is being done, the wonderful work that is being planned and done here are somehow out of the scope of what you will be looking into. So to some extent, the effort that is being done here to try to move some of novel initiatives into well-structured, organized, and funded clinical and well-controlled clinical trials, and then into standard of care are extremely, extremely important. That is work that we do not that we cannot do at NIH, although we would love to. So what I'm going to try to do here is to present to you some thoughts, or discuss with you some thoughts on possible strategies to enhance neurorehabilitation. Please feel free to interrupt me at any point, at any time, with questions, or we can go at the end. So one of the issues that we have been interested in is in trying to understand better stages of motor skill learning. And the reason we have been focusing on this question, my interest is mainly in rehabilitation of motor function, is that sometimes some of the interventions we apply and the measures we use are a little bit wider than we would like to, and the choices of endpoint measures are often unclear because our understanding of how we learn, for example, either a new motor skill or how we relearn a skill after brain injury, and the stages of learning are not so clear. So I'm going to review a little bit of this briefly. This is work that was done or reviewed by Diane, a fellow in my lab, recently. Whenever we are starting to learn to practice a new skill, there is a period of fast or early learning, which is usually what takes place the first time we encounter this new task. And the first time we do it, over the first hour or half an hour or day, there is a relatively rapid improvement in a skill that is often referred to as online learning, which means essentially within training gains, whatever performance improvements we experience during training. Then there are usually periods of time in which we do not train, and then we train again, for example, in this case on day 2. Now, and then repetitively over time. When we train repetitively over time, then we move from that initial early stage of learning into a slower slope of learning, usually referred to either slow or late learning. And at some point along the line, after multiple training sessions although this is debated, into some level of automaticity. Like, for example, the way we learn to ride a bike. This is something that we have with us, and then over life we sort of maintain, or at least a certain level we maintain. And this overall is referred to as retention of learning. Now, the important point is that with repetitive training sessions, there are two types of learning that I want to mention. One is online learning, which is within session performance improvements that takes place on day 1, day 2, etc. And then in between practice sessions, there are changes that develop in performance as well, and this is referred to as offline learning, and we will be talking a little bit about this as well in a minute. All along the process of learning a skill over the lifespan, there is what I would call multiple types of disturbances or disruptions that are completely different every day. That may, for example, when playing golf, could be the wind. On one day it's going to be more windy than another day, and that is to some extent compensated, or we compensate to that either consciously or unconsciously. Another day we are going to be tired. Another day we are going to have a certain level of pain. And this is what's trying to be referred to here as adaptation. That is, all over the process of skill learning, there is adaptation to a number of different elements, confounds that come up at different points. And this is something that is quite interesting and has been studied to some extent, less so in the process of skill learning, but it's something to keep in mind as well. Overall, adaptation to disturbances of different types is something that is considered at the time of looking at skill learning. There are clear neuroimaging correlates of learning in the early or fast period, usually characterized by increases in activity in the SMA and the dorsomedial striatum and the premotor cortex. But perhaps more importantly, whenever we start learning a task, there is a substantial amount of what's called error-dependent or error correction-dependent learning, and that is shown by, or is correlates with, increases in activity in the cerebellum. As we become better, a little bit better at the task after the initial stages of learning, what we see is that that error correction process in the late stages of learning stops to some extent or diminishes, and is related to a decrease in activity in the cerebellum with substantial increases in sensory motor, in activity in sensory motor areas and the dorsolateral striatum. So there are more or less clear neuroimaging correlates of early and late learning. And what I didn't mention about early learning is that attentional networks are very strongly active, are very highly active during early stages of learning, and a little bit less so over time. Now, the second concept I want to discuss with you is something that has been developing over the last 10 years. And I'm going to refer to this as a memory modification or reconsolidation. Whenever we learn a task, either in the motor domain or in the visual domain, training leads to formation of a memory. And as I was mentioning earlier, within session online learning. Now, that memory that we learn during training is consolidated or stabilized, usually offline, with the passage of time and or sleep. And we used to think that a consolidated memory is something that remains relatively stable, relatively constant. However, work done in the basic sciences and more recently in the clinical arenas, I'm going to show you in a minute, has shown us a completely different picture. The possibility that a consolidated memory can be, is modified, gets modified, and the fact that the consolidated memory is going to be changed. And not only that, the fact, as we will discuss in a moment, that it is feasible to intend to purposefully modulate, change consolidated memories. We'll get to that in a minute. How is that happening? Well, that's happening partially through the process of reactivation. This research has started to show that each time that we bring a memory to consciousness, that memory, through that reactivation, becomes susceptible to modification. We will get to that in the next few slides. So, the point I want to convey to you, this is work done by Nitzan Sensor, who's now back in Israel. When a previously consolidated memory is reactivated, it undergoes modification. And that modification could go in three different directions. Either stabilization of the identical memory, that memory could be enhanced somehow, or it could be degraded. This would be sort of a form of forgetting, if you will. So what I want you to do at this point, if you have enough caffeine in your system or not, is think for a moment that you could purposefully change a memory in your mind and just play as I'm talking with that idea and its possibilities. So this concept of reconsolidation or memory modification is quite, is going to becoming quite important. I'll give you an example of the type of information that we can start getting out of this. I was told to let people in the back know about, this is unpublished data, so that's why I was mentioning that to them. So I wanted to show you a little bit about this work to give you an example of something that may be interesting for in the field of rehabilitation. So motor skills, we know, are shaped through consolidation and reconsolidation processes. Each time we train, as I was telling you earlier, a memory and memories are modified. We know that rehabilitative protocols likely engage both processes of consolidation and reconsolidation. Each time a patient trains, he or she is going to take that memory that they formed in the previous training session, and we are trying to enhance it through training, but it's going to be modified in some way. We don't know exactly how. All we know is that the more the merrier in terms of practice. But there are more details to that. So The task in this study that was done by Nitzan and Ethan Bush was to have patients with chronic stroke who were able to perform some finger movements to type a sequence on a keyboard. They tried that sequence in a first day. Then they were tested on a second day. That is, whatever they learned on day 1 was brought back on day 2, and the memory from day 1 was reactivated. And then the effect of that reactivation was tested on day 3, on a third day. So the process from day 1 to day 2, or the process of changes in performance from day 1 to day 2, refers to as consolidation of the memory, and the one from day 2 to day 3 as reconsolidation or memory modification. So the patients were in general well recovered. I think that the most important point, these are chronic stroke patients, and they, as you can see here, and the lesions were pretty heterogeneous. And I think that the most important point I want to mention is that, as you can see here, the Figel-Meyer scores were pretty good. So what this slide is showing in the y-axis is the memory gain offline learning from day 1 to day 2. And you can see what's happening in stroke patients in blue and in elderly controls in red. So the magnitude of consolidation was more or less comparable. Offline performance improvements, 15 to 20% in both groups. Now see what happened with memory modification from day 2 to day 3. Essentially, what happened here was that the reactivation of the memory in day 2 in elderly subjects elicited additional offline learning from day 2 to day 3, while patients with chronic stroke showed a trend towards forgetting from day 2 to day 3. Certainly different from the elderly, and unclear and quite variable, as you can see, in terms of the magnitude of this effect. But clearly different between the two. So,
what's a normal young person
do this? Would do exactly the same. Probably a young person would be a little bit higher here than the elderly controls, but not a lot.
What's the difference between left and right, the young stroke?
We didn't have enough enough subjects to know that.
I would have expected a lot of variability.
And there is.
So, is there more
in relation to the, in this case. As you can see, the variance here is larger than this one. So we cannot say that there was forgetting. There was a trend there, but it wasn't quite clear. But it was certainly different than the other group. The variance was also was comparable in the case of consolidation, from day 1 to day 2. So why am I showing you this data? The reason I'm showing you this data is essentially to sort of bring the concept that in addition to the more training, the merrier, we don't know, for example, if, for example, instead of having a 24-hour period between day 2 and day 3, let's say that we have a 48-hour period, we could change this into a better memory, a positive memory modification. And that this issue of how do patients learn over repetitive training sessions and what changes within these days may be quite important, like the schedule or things like that. Okay.
I think a little bit of the concern here.
I'm sorry?
What is the area of the stroke?
Oh, yeah.
Yeah, because
so here you have the areas. I didn't put the slide with the maps, but this tells you in each of the cases.
maybe that's because that will affect the variability of the data.
Absolutely.
Yes. And Some areas are, more, are directly implicating memory, for example, other areas are not.
That's absolutely right, and you can see here, so the putamen and the corona radiata, the level of the lesions, most of these lesions did not affect directly pyramidal tract because otherwise the patients couldn't do the task. This is a selected group of stroke patients, and it's more, what I'm trying to raise here is more the issue of the importance of looking into these issues in general, because it's not memory as such the problem, because otherwise consolidation would have been different as well. Consolidation was fine. The problem was the subsequent memory modifications. They didn't keep going up as they could have. So there are a number of technical issues that can be considered at the time of looking into rehab protocols, but I want to show you something that is very simple, almost silly, I would say, but thought-provoking in my view, which affects what is really the gold standard in neurorehab, which is the ability to induce long-term retention of a skill. That's really what we are all shooting for. And we use a number of technological tools, but it's not only technology that can give us that. I'm going to give you a very silly example to some extent of how behavioral strategies can play a tremendously important role. This is work done by Mitsunari Abe, who's back in Japan now. He trained people in controlling with a force transducer held by the index finger and the thumb, a red cursor on a screen. So the more they press, the more the red cursor goes up, while there is a blue square that is moving up and down following a sequential order that's repeated over time. So we measured the error in the performance of the subjects who were instructed to keep the red cursor within the blue square constantly. And the distance between the two was the magnitude of the error. So the lesser the error, the better the performance. 3 groups of subjects were studied. 3 groups of healthy volunteers. All of them were paid to participate. One group, the one that received neutral training in this task, was going to receive $40 at the end of the study. One group, the rewarded group, started with $0 and were told that the better they performed, the more money they were going to make. Although we knew that it was going to be about 40 at the time of the experiment. And the punished group was told that they were starting the study with $80 and they were going to start losing money depending on how poorly they performed. Based on preliminary data, we knew that all of them were going to make about $40. So first, we determined baseline in the 3 groups. Then each subject underwent a period of 16 minutes of training in this task under rewarded, punished, or neutral conditions. And then performance in the absence of information, in the absence of reward or punishment, was tested immediately after training, 24 hours later, and 30 days later. And this is what we were interested in, to try to find, to determine if the conditions under which such a short period of training is performed could influence long-term retention of what was learned. And these are the results. This is error. So the higher the error, the poorer the function, and they improve over time, going to low error. This is performance in the 3 groups before training. Then they trained. They learned to a comparable extent immediately afterwards. But see what's happening over time. I don't want to get into too much detail here. Let's just focus on 30 days. So at day 30, in the rewarded group, performance was even better than immediately after training. And not a little bit better. I would say 30% better in the absence of training, while the other two groups, the neutral training and the punish groups, sort of tended to forget over the 30 days. Suggesting, in essence, that the framework in which we set up the training is extremely influential in what's going to be retained over time. This is, in addition to making me reflect highly on the way I educated my kids, in addition to that, sort of questions seriously the assumption with which I was trained that a patient with chronic stroke that is getting training should be happy because there's no training, there's no treatment. And just the fact of getting something matters, which probably does, but the conditions under which that is done is quite important. So I want to change gears to raise another issue here, which is, I believe, very, very important. When I discuss this issue of learning overall, I mentioned that we have performance improvements that take place during training, but in addition to that, there is an enormous amount of performance improvements that take place offline, either during sleep or just with the passage of time in between practice sessions. And this is likely to be very important because we know that stroke patients have serious trouble in sleeping. Sleep patterns are disrupted after stroke. And so this is something that might be influential as well. So I want to show you an experiment that was done by Sylvie Song, Sumbin Song, who's still in the lab, in which subjects learn a motor sequence, and it is possible when learning to try to utilizing different learning models to separate in terms of the stored skill what is called transitional and ordinal learning. This is a sort of complex psychological separation that is done, but in trying to make it clear, the simplest way to explain it would be that when we learn a sequence that is repeated over and over again without our knowledge that there is such a sequence, people are not told that, at some point we develop a form of unconscious knowledge that the key, for example, 2 is going to be pressed after the key 3. If we are asked consciously which is pushed first, we really don't know, so we cannot say it, but we do it. A form of, in other words, implicit learning, if you will. On the other hand, there is this form of ordinal learning which becomes more conscious, for example, knowing that we have to press the key number 2 in the third ordinal position within a certain sequence. That we can answer, yes, this is what we have to do. So it's a slightly different form or component of learning. Both are at play when we measure skill. And the point I want to make about this is that these different forms of learning do consolidate, are stored differently, online and offline, in relation to sleep. It is known that transitional representations improve with practice, that is online, while the conscious knowledge or ordinal knowledge improves during sleep. What this is telling us is that if we are trying to teach certain things to, or certain skills to a patient, and some of those skills are, must be consciously understood, must be consciously known, and be able to be reported as such, that type of skill, in the absence of proper sleep patterns, is going to get disrupted. While the transitional learning is going to be less affected. So this is something that is under study. So in other words, the importance of this point, the point I want to make here is that sleep matters, and how we handle the training protocols in relation to sleep modulation may have relevance in relation to the end results. So this slide, I only bring it up to make the point that there are really a very large number of treatment protocols that have been put forward to try to help patients with chronic stroke. Some involve peripheral nerve stimulation, FES, cortical or brain stimulation, stem cells, a number of different treatments, not many of which, or interventions, not many of which have become so far treatments. I'm going to start focusing now on brain stimulation and what we know about it. So there are different forms of brain stimulation. The two more common ones are transcranial magnetic stimulation, which is what's depicted in this slide, with a magnetic coil that delivers electric currents through induction of a magnetic field in relatively focal regions. And the other one is direct current stimulation that I'm going to refer to in a minute. The main point I wanted to make with this slide is that either with TMS or with TDCS, even when we are stimulating one region specifically, or at least targeting one region, the stimulation is not as focal as we would like to believe. Stimulation of a node in the network is likely to, and we know, spreads to a number of different interconnected networks. So most likely we are interacting with network effects rather than with single brain regions effects. When we stimulate the motor cortex, in this case, for example, with direct current stimulation, and this is work that was done in the lab by Janine Rice and Heidi Chambra. Heidi here is on the left. And this is a study that was done in collaboration with John Krakauer and Pablo Selnik when he was still in the lab. And what was done was test a group of subjects under practicing a motor task, a motor sequence task, over 5 days under the effects of anodal transcranial direct current stimulation of the primary motor cortex or sham stimulation. These are two different groups of subjects. And the point at the time was that the subjects who received anodal TDCS tended to learn more over time than the subjects that received sham stimulation, which is a result that has been replicated and has been described in many different laboratories by now. What was interesting at the time was that, for us, was that the effects of TDCS on learning seemed to be more as we will see in a minute, related to offline effects. And then Pablo has recently done a couple of very nice studies, one of which has been published, sort of following up or advancing, taking this work further. So the point here that I wanted to underline was that anodal TDCS did not modify online learning relative to sham much. So both groups learn about the same online during training. However, offline, the subjects that receive a nodal TDCS tended to learn a little bit more and clearly more than the subjects that receive sham. So offline performance in these individuals led to some level of forgetting, while in the group, a nodal TDCS group, some form of offline learning.
Which area?
M1, primary motor cortex. But again, this is the anode was placed over the motor cortex and the cathode over the contralateral supraorbital region. What that means is that we are targeting M1. Now,
was done during the task?
During training. During the training, yeah. So, but that doesn't allow us to conclude that the effect was due to M1 stimulation. So there is a caveat there. And then whatever performance improvements were present on day 5, sort of, or difference between the two groups remained present 3 months later. So maybe I'll skip this point. I just wanted to let you know that whenever we stimulate the brain, it is now feasible to monitor brain activity during stimulation in different ways, and particularly oscillatory brain activity, which is something that allows us to start studying more accurately the brain mechanisms that operate. Then I wanted to address this question, do we all respond to training protocols equally? Well, the answer just clinically is obviously no. But there are reasons why this is not the case, and there is a need to understand these reasons better. This is something that was looked at by Janine Rice and Britta Fritsch together with Bai Lu when he was still at NIH, in which we look at the influence of a BDNF polymorphism that is very prevalent, as we will see in a minute, in humans, because one every four persons, for example, 1 every 4 of us here in this amphitheater have this particular polymorphism. And the question that was put forward was, can BDNF influence the ability to acquire and retain a motor skill? And so this was an experiment that was done in parallel in humans and in mice. And the idea was to look at the effects of 5 days of training in that motor sequence task I just showed you, in human MET carriers and in Val-Val individuals. And mice performed the rot-a-rot task over 5 days again. And mice were either wild-type mice or MET-MET mice. And so the whole point of this slide is to show that in both models, 5 days of training led to better learning in the Val-Val humans than the MET carriers, and the same situation applied to mice, suggesting that certain, not necessarily this polymorphism, but either this or some sort of complex polymorphism configuration can induce different response to training protocols. And if so, this is something that we need to know, because it would allow us to individualize, to customize better and design better treatment protocols. Some people may need a little bit more training than others, or more intensive training than others, which is something that is being now studied in larger clinical trials. And that is a question that emerges from it. Now, I want to shift gears a little bit to be a little bit self-critical and critical about some of the how to look sometimes at this information that we are reading in materials. So when looking ahead, some issues that we need to consider, particularly in the brain stimulation literature, I think, is relate to the statistical analysis and interpretation of exploratory and hypothesis-driven studies, and know that they differ. That whenever we design a study and we power it to do a hypothesis-driven study, then we are going to be able to conclude that a certain intervention may be better than sham. But whenever we are sort of making a number of comparisons, only one of which is significant, for example, then the idea is that that data is quite important in order to provide information for future better designs, hypothesis-driven studies to target that particular thought. This is a confusing issue in the literature, which deals a little bit with this concept of matching between experimental design and paper and reports, written reports in hypotheses, and the idea of retroactive formulation of hypotheses because of the journal's pressure. So this is something that we need to start somehow dealing with as a community, and it's not clear. I think that it's interesting, something that helps a lot. This issue is what some journals are doing, and I'm going to maybe I'm mentioning that in the next slide. So this looks bleak, looks dark, looks terrible, but we need to bring some of these issues to consciousness to try to deal positively with them. So the issue of the true description of the number of comparisons in a way that doesn't hamper careers and publication possibilities. The issue that data mining is not bad as such. It's not a problem, but requires specific statistical techniques. The importance, whenever we get an effect in brain stimulation trials, of comparing effect size with accepted interventions, for example, which is not often done. And why are these problems? Well, one of the issues, I think, and this is completely open to discussion, if you will, is because of largely what I consider a publication bias. How to handle, for example, peer review and reporting of negative results in our community. It's very difficult. It's very complicated. But I want to bring up something that one journal is doing, and I have nothing to do with that journal, which is that whenever a trial is being proposed, then what you can do is submit to the journal the experimental design of the trial. That experimental design is submitted for peer review. And then the experimental design is accepted for publication or not. But if it is accepted for publication, then it's going to be published regardless of the results. And I think that this is an issue that is quite important, really, to sort of pursue. Wouldn't that make life much more reasonable from the investigator's point of view? The second point is that this concept of web-based data banks that are peer-reviewed, almost like a journal, if you will, that in a way that negative results can access the community rather than having to be hidden, so people don't repeat the same mistakes. Some of these issues should be moving to the influence as well, the issue of funding bias. Funding is largely sort of given on the basis of positive results. And again, the influence of reported negative results on career development and the influence on upcoming and published data. Yes?
Yeah, one of the issues in trying to publish in this, we're starting to realize is that the patients with stroke, of course, bring considerable variability. Matching them in different homes, etc. It still doesn't nullify or dilute the effects that are related to the tragedy of the lesion. Now, do you feel that there is a publication bias towards studies that are minimizing the variability to an extent where they're delivering a single session intervention, two-session intervention, maybe just as studies. But the studies that are delivering the long-term rehab are more prone to see variable gains in benefit because people are learning differently. And now if you add not just well-recovered patients, you add the big severely impaired patients, then all bets are off. So do you feel that reviewers would like to see cleaner data, but may, at times, trying to find the cleaner data may kind of negate the effect of really delivering a long-term protocol that may be a little more meaningful for long-term gains?
So I think that that reviewers are influenced by journals and by impact factors and things like that to the extent that they want the cleanest possible data, the sort of simplest possible data, the clearest presentation. We want to spend the least possible amount of time in reviewing because everybody has a tremendous pressure. So that is one aspect. But the second aspect is that I believe that reviewers need to be, the community has to be re-educated. So for example, on the concept of variability, the importance of variability depends on how you handle it, on what your hypothesis is. If your hypothesis is, and the conclusion you want to draw is that every stroke patient must be treated with 5 days of TMS at 1 hertz over the left M1, then you're going to be in trouble with that, regardless of what the result of 10, 20, or 30 patients are. If the hypothesis is that the goal is to relate variability in structural, in the microstructure of certain areas of the brain in relation to outcome of learning, then it's different because you are sort of relating learning with the integrity of one particular brain region, as somebody was asking a couple of minutes earlier. So a lot depends on the claim. That said, there is I would say, a knee jerk in reviewers that even before or even after they read that aim, they are still having, or we are still having sometimes, the knee jerk of saying, I want a more, this cannot be studied in stroke because patients are heterogeneous. So it's necessary to state up front very clearly, because everything is so variable, we are looking for an intervention that has a meaningful size effect that could overcome that variability. In order to do that, we need the variability. We need to embrace variability. And definitely anything that's longer term and more significant number of patients is going to be much more important than 5 days that is meaningless other than hypothesis generating. So anyway, so these are some, I'm concluding a little bit with raising these issues because I think that to some extent it is important as a community to start embracing these problems, to start reporting these problems. And for example, sometimes when I review a paper or papers, what I say, I judge the paper, and then I ask the authors to provide this information in supplementary data and so on, regardless of the outcome of the review or something like that. It's a matter of education even. And to try to to make it as clear the information as possible. Now, even with all these problems, the reason I brought this slide, which is the sun rising in Machu Picchu in Peru, is because this is a field, the field of brain stimulation to modulate performance is starting, it's growing up, it's developing, it's extremely exciting. And these are issues that are extremely exciting to face. And this is the stage we are in. 5 years ago, 10 years ago, 15 years ago, we were in a different stage the problems were different, and they were they were at the time overcome. So now we have these problems, we need to overcome them, and we will with everybody's help. So I would stop here. Yeah.
When you do transcranial magnetic stimulation to a stroke patient, how common is to see side effects like seizures, that kind of side effects?
So there are a number of different forms of brain stimulation. So for example, with direct current stimulation, to my knowledge, there has not been any seizure reported in the literature. With TMS, there have been under safety, under current safety rules, maybe a couple in the history. There were more at the time when the safety tables were being generated. At the time, there were 4, 5, 6. That said, maybe the total now is probably 10 to 15. If somebody has more updated information, please let me know. But in general, it's minimal. That said, stroke patients do seize. Stroke patients, about 2, 3, 4, 5, 6% develop seizures, regardless of any stimulation. So it's even hard to know when it happened, if it was related or not.
And other side effects?
For TMS, some headaches, depending on the stimulation intensity, local discomfort. For TDCS, I would say essentially skin. There may be skin irritation because of the electrode, which is like an EKG electrode, like when you have an EKG and you pull the electrode out. Yeah.
On the sleep study for memory stimulation, that's interesting to know. Is there a particular stage of sleep where this is? Do you monitor that? And the other question is, if you were to interfere with sleep, is that really?
So the answer to that, I have the same questions you have, so we are starting now a couple of sleep studies trying to address those questions. That said, there are 2 investigators that have looked into these issues to some extent in detail. One is Jan Born in Tübingen, and the other one is Tononi, Giulio Tononi, that have looked into these issues nicely, and we are sort of using their information to design our trials. Though that is crucial information that needs to be acquired.
You don't know the answer.
I don't know. Yeah. Yeah.
Along those same lines, you know, there's plenty of drugs out there to help induce sleep. Has there been anybody looking at how the drugs affect the learning that occurs during sleep?
Probably there are studies about that in relation to in the clinical rehab literature. I haven't followed them and I haven't looked at that yet. I'm at this point more interested in understanding the sort of which sleep stages are the ones that matter, because then I would go to the question of what do the drugs do on each of these different stages. So I haven't done it, but I'm sure that there should be literature on that. Yeah.
So I don't have the answer to that question, but I can tell you, in our stroke rehab unit, it seems to me we do things that's convenient for the providers. So we take vital signs at midnight, we draw blood at 3 in the morning, we turn the lights on, all the nurses talk, keep the patients awake, and all the patients are kind of, half asleep. Essentially, of course, I come at 7 in the morning to wake them up. Well, I wonder how much iatrogenic negative learning happens in our rehab units because we don't have a real good appreciation for the sleep issue.
Yeah, that's why. So again, this talk was more designed to be thought-provoking than telling you in the stroke unit you have to do X, Y, Z. But at least if we think about it, then we will be seeing trials coming out, giving us more information. I agree, it's extremely important. So when I saw this data, so this study was not done thinking in stroke. This study was simply done because the fellow was interested in looking into these two forms of learning. And then when I saw the results in normals, then I sort of thought, well, exactly, I sort of thought exactly what John mentioned. Yeah. Next.
So you showed a slide early on that during early learning activity in M1 goes down. That was one of the areas that was shown. And I just wonder what the magnitude of that decrease is and the time.
Right. So, so this is a controversial, somewhat controversial issue, but in the end, I think that that is the common knowledge. In the very beginning, whenever we do a difficult task, in the very beginning, we are sort of assigning a number of brain resources. We are first time we play golf. What is this? How do I move it? I mean, M1 is bright, everything is bright at that particular point. As we learn in that, at the end of the day, of the training day, the same movement that in the beginning make the brain put the brain on fire can be done actually with a relatively smaller activation, an economy principle, if you will, at play. Now, the magnitude of that depends on the change depends on the complexity of the task. If the task is simple movement, then it's going to be a little activation very early on and a little bit less afterwards. If the task is a complex sequence that the subject is learning, there's going to be a very active region that's going to decrease substantially 40–50%, if not more. With the caveat that we can never say in fMRI really what's the real space.
Well, I'm just curious because a lot of brain-computer interfaces report from M1, and that may be one kind of brain places report from, but that's not where the learning is occurring.
Well, no, the learning is occurring. So the within session or within training, learning, the first day is not necessarily super reliant across different learning protocols in M1 alone. But over time, or in the long term, M1 is very strongly involved and gets more and more active. That's why I showed in the slow period or in the late learning period, the M1 activity keeps increasing.
Excellent talk. Thank you, Dr. Cohen. You mentioned coupling recording oscillatory activity with TMS.
TDCS.
Or TDCS. Okay. Let me ask you hypothetically, coupling oscillatory recording, specifically looking for maybe theta rhythm, which is known to both the hippocampus and somatomotor cortex, the laying down of long-term memory, of motor memory. If you were to apply TMS only during peaks in the theta rhythm, if it would perhaps enhance laying down of long-term motor memory.
I mean, that would be an excellent hypothesis to look into. I think that the main problem there is that we cannot do with MEG. Well, you can do with EEG, sort of look into this oscillatory activity and then determine which the predominant frequency of oscillatory frequency during a learning task for that individual, because it's different from individual to individual, and then entrain that frequency purposefully with either TMS or alternating current tags, alternating current stimulation. Yeah, that's something that is being, there is some information in the literature, maybe one or two papers that are useful, but it was not done as you think. Yeah.
I know there are a lot of questions, so Dr. Cohen will be down here, but some of you do have to leave, so I'm going to stop our our presentation.
So I'm going to give Dr. Cohen this book. So Cheryl gave this to me, and this is pretty cool. You know why? Because it says here, we often hear that nothing seems to ever happen in Cleveland. And, you know, we get a pretty bum rap, right? The records prove differently. Perhaps those of us who are transplants, and I think most of us are transplants, I think, find more to praise and love about this great city than Clevelanders. And I think that's true. We love this city, and thank you for coming.
Oh, thank you very much. Thank you, John.